We are going to talk about cell, and this is the part one of cell that we are going to talk about. These are the learning objectives that we are going to learn about cell in part one. You can pause and read through. So let's start with what is cell as we have in our learning objectives. Cell is the basic structural and functional unit of life. That means it is the least form in which life can exist. Life doesn't actually start from cell. We have other structures which combine to give us this cell. Life starts with atom. These atoms like nitrogen, like oxygen, carbon, they combine to give us molecules. Molecules like deoxyribonucleic acid, DNA, glucose, amino acid, fatty acid, and glycerol. These molecules they combine to give us what we call organelles. These organelles they combine to give us what we call cell. It is at the cellular level that life actually exists. These organelles they are not living organisms. Organelles like what? The nucleus, the mitochondrion, ribosome, endoplasmic reticulum, lysosome, vacuole, etc. These organelles are not living organisms, but when they combine together, they give us something that is living. For example, as we have in amoeba, amoeba exists at the cellular level. We have paramecium. Paramecium exists at the cellular level as well as Aglena. So we have example of cells that we have in human. We have nerve cell, we have muscle cell, we have sperm cell, which is the smallest cell that we have in human, and we have ovum. The ovum is the largest cell that we have in human. In plants, we have cell like parenchyma cell. The food we eat, most often they are made up of this parenchyma cell, like the mango, the oranges, etc. We have collenchyma and sclerenchyma. The parenchyma, collenchyma, and sclerenchyma, these are cells that are found in plants. In some cases, where we ask you to give example of cells found in human, or example of cells found in plants, or example of organisms that exist at the cellular level. So all of them they are important in different ways. Next we have is the history of cell theory, how this cell theory came about. It all started in 1665 by Robert Hooke. Robert Hooke, he observed cork from the back of an old stem. The cork is not a living organism, it is non-living. They are dead cells. He observed this cork under a microscope. A microscope is an instrument used to magnify objects. He observed this cork, and what he saw a small box-like structure, and he named these box-like structures as cells. So if you are being asked who gave the name cell, it is Robert Hooke that gave the name cell, and Robert Hooke is being considered as the father of cell theory. In 1670, another scientist called Anthony van Leeuwenhoek. Mind you, you're supposed to know the correct spelling of the name of this scientist. Anthony van Leeuwenhoek, He improved on the microscope that was being used by Robert Hooke. The microscope that Robert Hooke used was just able to magnify an object 100 times of its original size. It is Anthony van Leeuwenhoek that improved on the microscope, which can increase the size of an object 300 times of its original size. He observed a living cell, which he called animalcules. And these animalcules, they are what we know as today as bacteria and protozoa. So what Anthony van Leeuwenhoek was living, but what Robert Hooke observed was not living. In 1835, Flex Dujardin discovered the living part of the cell. Now we said Robert Hooke observed a dead cell, and Anthony van Leeuwenhoek observed a living organism. Now there was a sense that a living organism is made up of living parts and is made up of a dead part. As Robert Hooke observed a dead cell from a plant that was living, and Anthony van Leeuwenhoek observed a living organism. Is it that all living organisms are made up of something that is living and something that is dead? So it is Flex Dujardin that discovered the living part of the cell, and he named this living part as sarcode. But today this sarcode is known as protoplasm. This name protoplasm was being named by Johannes Purkinje. Johannes Purkinje, he gave this name protoplasm in the year 1861. The protoplasm, it consists of two things: cytoplasm and all its contents together with the nucleus. Protoplasm equals to nucleus plus cytoplasm. Since we know that living organisms, they consist of cells, so Matthias Leiden went further to conclude that all plant tissues are made up of cells. A year later, 1839, Theodore Chewine, a zoologist, Theodore Chewine extended this idea to animals, and he discovered that all animals are made up of cells as well. This gave us the two cell theories, that one, cell is the basic structural and functional unit of life, and two, all living organisms are made up of cells, because by then we are just having kingdom Animalia and kingdom Plantae, and both are made up of cells as stated. So in that case, they came up with the second principle of cell theory, that all living organisms are made up of cells. So the first principle is that cell is the basic structural and functional unit of life. And the second principle is that all living organisms are made up of cells. In 1855, Rudolf Virchow, he added the third principle, which he stated as omnis cellula e cellula, meaning that every cell comes from pre-existing cell. So he completed the cell theory. So let's talk about the three main principles of cell theory. We have the first principle, which is being stated here as all living organisms are made up of cells. Number two, cells are the basic unit of life. And the third one is that all cells came from pre-existing cells. That means they are coming from cells that exist before, not cells that are dead. Because there was a debate among scientists. Some were claiming that living organisms came from non-living organisms, and some stated that living organisms they came from another living organisms or something that was living, not something that is dead. And the others were claiming that living organisms they came from something that is living. It is Rudolf Virchow that ended that debate in his experiments, in which he stated after the experiment, omnis cellula, e cellula, meaning all living cell came from pre-existing cell that we are living. So after we have defined cell, we have talked about the history of cell theory, we have talked about the principles under it. Now let's talk about types of cell. So based on the number of cells, either we have one or more than one, we have two types of organism. We have the unicellular organisms. Uni mean the same, or it can also mean as one. So in this case, these organisms are made up of only one cell. For example, amoeba, bacteria, paramecium. These organisms, they are just made up of one cell. And we have multicellular organisms. Multi, or sometimes they pronounce it as multi, means many. That means these organisms are made up of many cells. Example, humans. In fact, humans, they are having trillions of cells. So in terms of cells, you are rich, you are a trillionaire. And we have monkey, cat, mango, rice, etc. They are all multicellular organisms. On the basis of the nucleus, we have three types of cells, or three types of organisms. We have eukaryotic cells or eukaryotic organisms. When we talk about a, when it precedes before a word, most often it means not. So in this case, the cell or the organism, they are not having a nucleus. If you are being asked what is an eukaryotic cell or cells, you can say that these are cells that do not have a nucleus. They are otherwise known as non-cellular organism. Example we have is a virus. Below here, we have a bacteriophage. A bacteriophage is a virus that causes a disease to bacteria. In other words, they infect bacteria. Just like bacteria, some of them, they are causing disease to us. Virus also, which we call bacteriophage, they cause disease to this bacteria. So while the bacteria, the parasitic bacteria are causing disease to us, virus is also causing a disease to them. And the name of that virus that causes disease to bacteria, I said they are called bacteriophage. And this diagram here is a typical example of a bacteriophage. Now this bacteriophage is having DNA or RNA as its genetic material. It cannot have both. They can either have one of them but not both of them. If they are having DNA, they cannot have RNA. If they are having RNA, they cannot have DNA. And the nucleic acid is being surrounded and protected by the capsid. This nucleic acid, it contains the genetic material. It is the one that has to be transferred from the parents to the offspring. And the capsid, it is what protects the nucleic acid. And we have the finger here. This finger here, it is there for attachments and transfer of the nucleic acid into their host. And the other important part we have here is the contractile sheet. This contractile sheet, it is the one that compress in order to inject the nucleic acid, that is the DNA or RNA, into their host. Now let's repeat the structural parts of a virus and their function again for more understanding. We talk about the nucleic acid. We say it contains the genetic material which has to be transferred to the offsprings. And we have the capsid. As I said, it is a protein coat that protects the nucleic acid. And this capsid here, it assists in the attachment of the virus to the host cell. So just observe here, this red structure you see here, this is the nucleic acid, and the blue structure you see surrounding this nucleic acid, it is the capsid. And we have the envelope. The envelope, it is not found in all viruses. That's why we have on the slide if present. This envelope, it is a lipid layer. And this envelope, it is being derived from the host cell. It is not the virus that has to create this lipid layer, this envelope that is being surrounded with. It is from the host cell that the virus derives this lipid layer, and it aids entry into the host cell. And we have the spike, which is a glycoprotein. That means it is made up of carbohydrates and protein, not like capsid that is just made up of protein. The spike, it is made up of protein and carbohydrates. So this spike, it is a projection on the envelope. And we said the envelope here, it is not found in all viruses. So if this spike is found on the envelope, that means even the spike is not found in all viruses. This spike, it enables the virus to attach to and penetrate the host cell. And we have the tail fiber. This tail fiber, it is responsible for recognizing. So it is the one that has to recognize that this is our host, this is not our host. Because not all living organisms that a particular virus can infect. It can infect some and some it cannot infect. So it has to recognize its host, the host that it can live comfortably in, or the host that it can cause disease in. And we have the contractile sheet. Just like I said, we have the contractile sheet in a bacteriophage, virus that cause disease in bacteria. So what the contractile sheet will do, it will contract in order to derive the T to this finger here through the host, and it facilitates the injection of the viral nucleic acid into the host cell. In some cases, people consider virus as a living organism. Some do consider virus as a non-living organism, and others consider virus as a bridge between a living organism and non-living organism. So after this explanation here, you can decide wherever you want to be, whether as living, non-living, or in between the two. So let's take the reasons why viruses are being considered to be a living organism. So one, they can reproduce within a host. Reproduction is one of the key defining feature of living organisms. So if viruses can reproduce, that means they are living organisms. Non-living organisms, they cannot reproduce, like stone, sand, they can't reproduce, never. And they contain genetic material. The genetic material is found only in living organisms, and it is the one that has to transfer to the offsprings. So therefore, if viruses are having genetic material, we can consider them to be a living organisms. Let's look at the other side of the coin, in which some agreed that viruses are considered as non-living organisms. So their reason were as follows. One, they live and reproduce only when they are in a living organism. Even viruses are reproducing, but they can only do that when they are in their host. They cannot reproduce outside a living organism. So in that case, must consider this virus as non-living organisms. And two, they are acellular. That means these viruses, they are not having the complex structure that is found in other living organisms. For example, they are not having mitochondria, they are not having a nucleus, they are not having ribosome, they are not having endoplasmic reticulum. There are many organs that they are not having. The third one is that they can be grinded into a crystal form. Living organisms, when you grind them, they form something like a paste or something like a mold. When you grind something like a stone, you can observe they can be grinded into a dust or into a powdered form. So a virus can be grinded into a dust or a powdered form. Then viruses are being considered as a non-living organism. So now let's talk about some example of viruses and the disease that they cause. Let's talk about the common one that we know. We have Ebola virus, causes Ebola. We have monkeypox virus, causes monkeypox. Coronavirus causes coronavirus disease. We have hepatitis virus, cause hepatitis. We have human immunodeficiency virus, cause acquired immunodeficiency syndrome. Cause acquired immunodeficiency. Now let's talk about prokaryotic cells. So prokaryotic cells, these are cells that do not have a true nucleus. That means they are not having the nuclear material being surrounded by a nuclear membrane. They just have this nuclear material called nucleus scattered within the cytoplasm. And from here as well, we are going to talk about this structure because you will be asked to label them. And we are going to explain the function here and we are going to repeat it in the slide coming. So you can listen here and listen next and understand more. So number one, we have the capsule. This capsule, it is the outermost layer here in bacteria. This capsule prevents the bacteria from desiccation. That means it prevents the bacteria from drying up. It also protects the bacteria. And we have the cell wall. The cell wall, it is also there for protection. It gives shape to the bacteria and it allows free entry of substances. While the cytoplasmic membrane, also called the plasma membrane or the cell membrane, this plasma membrane or cell membrane, it is also there for protection. It is selectively permeable, not like we have in cell wall that is freely permeable. That means it can allow all substances to pass through. But the cytoplasmic membrane, not all substances are allowed to pass through. Some will be allowed to pass if they are needed by the bacterial cell, and some will not be allowed to pass. Next we have is the pili. This pili is having two functions. One, it helps the bacteria in attachment. Two, it helps in the transfer of gametes between bacterial cells. And we have this whip-like structure called flagellum. This flagellum, it is there for movement. And we have the ribosome. The ribosome, just like we have in any other organisms, it is there for protein synthesis. But in here, we don't have mitochondrion that is there for the production of energy. In this case here, the production of energy, it happens within the cell membrane of the bacteria, which is being labeled here as cytoplasmic membrane. Now let's talk about the part of the bacteria and their function as we have described it before. We have the cell wall that provides shape and protection to the cell. We have the plasma membrane or cell membrane that regulates the entry of substances within the cell. That means it enables substances to enter into and out of the cell. And it is the site for energy production. As I said, bacteria are not having mitochondrion. Next we have is the cytoplasm, which is a gel-like substances that is found within the plasma membrane, and it is where the metabolic reactions or chemical reactions happen within the cell. And also all organelles, they are being suspended within the cytoplasm. And we have the nucleus. As I said, bacteria are not having nucleus. What they are having is just a nuclear material that is found floating within the cytoplasm. And that nuclear material that is found floating within the cytoplasm, not having a membrane, it is called nucleate. And we have the ribosome, just like in any other cells in which you can see a ribosome. The function of the ribosome, it is responsible for protein synthesis. And we have the flagellum, that whip-like structure in front of the bacterial cell. As we have it in the previous slide, it is responsible for movement, also called motility. And we have the pili or fimbriae, which is a short hair-like projection that is used for attachment to the surface. But there is a difference between the pili and the fimbriae. Both help in the attachment of the bacteria to a surface, but it is only the pili that can help to facilitate the exchange of genetic material between two bacterial cells. And lastly, we have the capsule, also called glycocalyx, a slim layer that protects the cell from desiccation, and it helps in adhesion. That means for the cell to attach on a surface or a host. And we have the plasmid. This plasmid, it is optional. It is not found in all viruses. It is the extra chromosomal DNA molecules. So bacteria, they have their main DNA molecules and they have an extra DNA molecules called plasmids. The plasmids, they carry additional genes, and these genes, They are responsible for antibiotic resistance and other functions. So below here we also have bacteria and the disease that these bacteria are causing. We have one, Clostridium tetani. Make sure you know this spelling correctly. If not, you will not earn any mark. And we have Neisseria gonorrhea, which causes gonorrhea. Clostridium tetani causes what we call tetanus. Vibrio cholerae causes what we call cholera. Treponema pallidum causes what we call syphilis. Know this bacteria and know the disease that they cause, and don't forget their spellings. Next we have is the importance of bacteria. So here we are going to talk about importance of bacteria and we are going to talk about their functions. One, they play an important role in breaking down of dead organic matter. For example, if you have an old bread, you can see something brownish coming from that old bread. That is what we call the bread mold. While in some cases, they might come with an bread and they tell you this is an old bread and they put something on it, and that something they put on it you will be asked to label. That is the bread mold. It is a bacteria and they can ask you many questions under it for you to answer. But all the questions will be based on an idea on bacteria. Two, they contribute to bowel remediation. We'll talk about bowel remediation. We are talking about the cleaning of the environment. So as they are breaking down that dead organic matter to return the nutrients back to the soil, they are helping to clean the environment. And they are used in food industry for fermentation. So fermentation is the process of breaking down an organic matter in the absence of oxygen. So these bacteria, based on that idea, they are used in the production of yogurt, cheese, and beer, etc. Number three, they are utilized in the production of vaccine. When they want to make vaccine, they use the bacteria itself that is causing that disease. They mutate it so that it cannot cause any disease and they inject it into a healthy human in the form of what we call vaccine. And they are used in the production of insulin. This insulin, it is there to regulate the blood glucose level. And also we have some beneficial or useful bacteria that can compete with harmful bacteria in the body, like the one we have in our gut or in our GI tract. They compete with harmful bacteria in terms of space, in terms of nutrients, thereby helping our immune system to defend against infection. And lastly, as we have on the slide, these bacteria, they form symbiotic relationships with plants. Some of them, they enable some plants to take up nitrogen directly from the atmosphere. Example of this bacteria we have is Rhizobium. And lastly, and the one that is very common of their economic importance is that they cause disease in other organisms. When we talk about economic importance, it doesn't mean good, good, good. It can be bad. You can also say that they cause food spoilage. That one is also another economic importance.